The thylacine, more commonly known as the Tasmanian tiger, was the largest carnivorous marsupial at the time of European settlement in Australia. They were once common throughout mainland Australia, but became isolated in Tasmania after flooding of the Bass Strait and the spreading of dingoes on mainland Australia. Figure 1b shows that the thylacine was widespread, however did not occupy the western mountainous regions. It preferred dry eucalypt forest bordering open wetlands and grasslands in which they hunted. The thylacine was feared from the beginning when Europeans arrived in 1803. With them they brought sheep, and it was believed that the thylacine was a threat to the sheep. As a result, the government brought in a bounty scheme, which paid £1 for every thylacine killed. This scheme lasted from 1888 to 1912 and saw the slaughter of 2,184 thylacines. In figure 1c you can see the collection rate of thylacines, as well as the steep decline in collections from 1905 to 1908. The last known wild thylacine was killed in 1930, whilst the last captive thylacine died on the 7th of September 1936 in the Hobart Zoo. An article linked to genetic diversity preceded Extinction of the Tasmanian tiger was published on the 18th of April 2012 by scientists Brendan Menzies, Marilyn Renfrey, Thomas Hayder, Freda Mayer, Thomas Heidenbrandt and Andrew Pask. Their research was aimed at determining if low genetic diversity aided extinction of the thylacine. This scenario can be seen with the closely related Tasmanian devil. It has been found to have low genetic diversity, which may have contributed to the spread of the fatal tumour disease. To determine genetic variability, the team analysed the hypervariable region in the mitochondrial DNA control region. Previously, 2 individual thylacines had their DNA sequenced. These 2 sets of data, along with an additional 12 specimens, were used. Table 1 shows the sequence length, location, date collected, sex, tissue, polymorphic sites of the individual specimens. Figure 2 shows the variability between the different DNA fragments and the location of these fragments in the DNA sequence. There are a total of 16 fragments ranging 11 locations. The 11 fragments below in blue represent indistinguishable sequences ranging from 100% to 99.7% identical, whereas the red fragments above represent sequences that are less identical, ranging from 87.8% to 96.2% identical. During the investigation, there were only 6 nucleotide differences between all the specimens sequenced. These findings support the team's theory that the population of thylacines was suffering from low genetic diversity prior to their extinction.